In 7.110, IB would like you to be able to be familiar with some of these molecular visualizations and play with them to try to get different images and pictures, because it's quite a common tool that scientists use, and some tool that you might want to use in an extended essay or in your internal assessment. So this is a nucleosome. So let's just talk about what a nucleosome is first. So a nucleosome is the combination of DNA plus protein. So it's those two things together. And that's what your DNA is. If you remember way back in 1.3, I think it was, that prokaryotes have naked DNA, which means no proteins, and eukaryotes, like us, we have something called chromatin, which is a combination of DNA and protein. Now, I think it's pretty obvious like what the DNA is in this image. You can see the double helix, right, all around the outside here. And on the inside is supposed to be the part that's protein. Now, if you look at this, you can see spirals in there as well, right? This green one has that spiral shape, yellow, they all do, on the two different colors of blue. That's because, right, the secondary structure of proteins, one of the secondary structures is called an alpha helix. So that's what you're seeing there. I'm going to draw what this looks like first, maybe in like a simple model, like a cartoon model. So there are 8 protein beads that are stacked like right next to each other, like this. So if you can picture, like these other ones are kind of behind it, and these are in the front. Not the greatest drawing, but there's 2 in the back, 2 in the front, these 2 are in the back, and 2 in the front. So that's what these colored, the yellows, the light blues, the greens, the blues. So you can see like maybe there's 2 green ones here, 2 yellow ones here, 2 blues here, and 2 light blues here, for a total of 8. Now, if we could rotate this, we could see this better. In this image, we're looking from the top, so it's kind of hard to see the different layers as I've drawn it. And then, actually, I'm going to erase this real quick so we can see it better. So wrapping around our molecule twice is our molecule of DNA. So we have the double helix coming this way, and then going behind, and then coming back on this side again. That kind of makes sense. And then I'll just make it a double helix to the best of my ability. And then comes around on the other side. Something like that. It wraps around. So that'd be the DNA wrapping around twice. And if I could rotate this, you'd be able to see that underneath this, and you can kind of see it, underneath this, there would be another strand of DNA. The third part, there's 3 parts to this model. The third part of a nucleosome is something called the H1 protein, which I'm going to represent in green. So this gives it structure and stabilizes the whole complex. Here, the H1 protein would be down here. So in summary, a nucleosome is made up of proteins called histones, and there are 2 types of histones. There's this H1, which I drew here on the right, there's that one, and the other histone, which I've drawn in red, are called the histone beads. So those are our 2 types of proteins that make up this nucleosome. And the last part is the DNA. So we also have our molecule of DNA. And together, they make a nucleosome.